Hey everyone, it's Ahem and Manvi here reporting to you on the topic of human cloning. Let's start off with, what is human cloning? Human cloning is essentially a genetic copy of an existing individual. You know, like two of me's? The science behind cloning is that you're replacing the nucleus of an egg cell with a nucleus containing the DNA from any of the millions of body cells of an individual. Thus, you produce an embryo leading to a normal birth of an individual that is a clone of the original parent. It's a very complex process, but by the end of it, you'll have multiple copies of an individual. Wow, imagine that, a world filled with multiple hems everywhere.
There are two types of human cloning. The first one being reproductive cloning, which produces a copy of an entire human which will be genetically identical. This process employs using a technique that is referred to as somatic cell transfer. The second type of cloning is therapeutic cloning, which is a process that produces embryonic cells to replace injured or destroyed tissues in the body.
Okay, before we get more into detail on cloning, let's take it back to where it all began. Mammals were the main animals used in reproductive cloning. In the 1960s, John Gordon combined an egg cell, in which the nucleus was removed, with the nucleus of an intestine cell to clone frogs. This did result in the cytoplasmic material to be transferred, but the tadpoles did not live to maturity. Then we fast forward 15 years. Scientists Salter and McGrath performed nuclear transfer to make the first cloned mouse. In this case, Salter and McGrath used a mouse's zygote acceptor cell, in which the cytoplasm was not touched but the nucleus was removed, and the donor nucleus from a genetically distinct mouse embryo. This resulted in a positive outcome as many clones had developed into adult mice. Fast forward one last time to 1996, where the Rosalind Institute in the United Kingdom had successfully cloned the first mammal sheep named Dolly. Nuclear transfer was used, in which the genetic material from an adult female sheep was transferred into an egg, where the nucleus containing genetic material was removed.
The growing embryo is used to manipulate that cell undergoing mitosis, in which this all revolves around recombinant DNA technology. Recombinant DNA technology is where you're altering genetic material outside of an organism to obtain enhanced and desired characteristics in living organisms. Thus, you're isolating and characterizing genes and inserting a specific DNA gene sequence that is needed.
Exactly. Cloning is a long process in which you are isolating a particular sequence of DNA from a complex mixture of different DNA sequences. To clone a fragment, you need to insert it into a plasmid, which contains a cloning site where the foreign DNA can be inserted. A gene that destroys antibiotics, like ampicillin, is inputted to allow selective growth of the host cell and a replication organ to allow the plasmid to replicate in the host cell. To insert foreign DNA, a restriction enzyme needs to cleave the plasmid DNA at specific sites. Foreign DNA is then inserted into the cut, which then contains the cloned DNA fragment. In plain English, you're cutting and pasting exactly what you want.
In the process of somatic cell nuclear transfer, the nucleus of the cell, which accommodates the organism's DNA of a somatic cell, is extracted and the rest of the remaining cell is discarded. While that is happening, the nucleus of an egg is removed and the nucleus of the somatic cell is inserted into the egg cell. After being inserted in the egg, the somatic cell nucleus is reprogrammed by its host cell.
It involves the process of somatic cell nuclear transfer as well as parthenogenesis. Parthenogenesis is the asexual reproduction in which the growth of the embryo happens without fertilization. Thus, there is no mating required. This is still theoretical as there has not been any experimentation because it is believed that the uterus of the mammal needs to have a fertilized egg attached in order for the offspring to be produced.
The egg now contains the nucleus of a somatic cell which is stimulated with a shock and starts to divide. Human cloning has many benefits, such as it has the ability to eliminate defective genes when healthy human cells are reproduced. It can also cure some disorders by replacing any damaged tissues. It assists in faster traumatic recovery. Human cloning also gives parents the advantage of choosing particular traits for their children, ranging from gender, eye color, and other characteristics. This new technology also has the ability to eliminate infertility within women. It can also destroy viral epidemics and can lead to advancements within organ transplants.
Human cloning is also seen as unethical because there are a lot of risks that come alongside artificial creation. Human cloning interferes with nature, and it goes against some religious morals and ethics. Since human cloning is still experimental, there is a possibility for there to be abnormalities in offspring. Adding on, due to cloning being an almost identical copy, this can cause a monoculture, which can lead to psychological distress. But the question at hand is, would the cloned individual have the same rights as the original? In conclusion, we believe that cloning is a new technology in the world of science that needs to come to light. Each and every day, there are new discoveries being made, diseases being cured, and if cloning is able to aid in research to help better our world, why keep it in the dark? These were just our thoughts and opinions, but after listening to this, what do you think?